Good morning, sir. Good morning to all. Sir, my voice is audible?
Yes, good morning, Maheshwari. Your voice is pretty much audible. Yeah.
Yeah. Students, good morning, students. Please inform to your friends, let them join as early as possible. YouTube Live and Facebook Live is also going on. Who are facing in airmid in issues, you can join YouTube or Facebook. Okay, thank you. Sir, you can continue.
All right. Thank you, Maheshwari. Let me share my screen and let me know once you are able to see my screen. Okay. Is my screen visible?
Yes, sir, it is visible. You can continue.
All right. So thank you. Very good morning to all of you, and once again, warm welcome to all of you for this online training organized by Andhra Pradesh State Skill Development Corporation. And The topic we have picked up is drug discovery and development and overview. And in last close to around 10 days, we have covered different phases of drug discovery development, role of in vitro experiments, cell-based models, alternatives to animal experimentation. Then we have discussed about importance of absorption, distribution, metabolism, excretion, ADME of drug candidates early in preclinical drug discovery. Then we have talked about toxicology studies, and we have also talked about in vitro assays of ADME as well as in vivo testing system. So then yesterday we have talked about specific handling on animals. What are the welfare ethics about CPCSEA, the regulatory agency who oversees the experimentation on animals in India, and about the dose administration, different routes of administration, about the blood collection. Those are the things which we have covered so far. And today we will. Yesterday we have stopped at GLP, I guess. So I think we will take up from there. So today we'll do a quick recap, not the elaborate recap as we used to do on a daily basis, but we will just restrict our recap to just few slides, 3 to 4 slides, as a summary slides from our yesterday's discussion, and then we will take up the topic of good laboratory practices, which we started yesterday. Okay, so since today's topic is majorly it's about the good laboratory practice, like in order to do good science, what exactly you need to do. That is what how a lab should operate, and how you can really generate quality data and authentic data, reproducible data, reliable data from any experimentation. So in order to do that, we have to follow good laboratory practices. So we will cover in detail about GLP, and since it's not that scientific a topic, but it's very important from drug discovery and development point of view, because when you go for approval and submission of data to regulatory agency, of course they want that certain amount of data should have been generated as per the principles of GLP. So it should be GLP compliant. So it's very important to understand the role of GLP and how it can be maintained. And of course, there are a lot of misconceptions in people's mind about GLP. But once we complete this presentation, you will realize that it's very easy. It's not that difficult. Only thing is you need to be organized. Everything, whatever we do, should be documented. So it's all about documentation, and not only from a GLP perspective, but if you follow these good practices in your own lab, in your college, in your university, in your contract research organization or institutes, the quality of science would be warranted. In the sense, you will, I can guarantee that whatever science you will do, it will be a good science if you align yourself to good laboratory practice principles. Okay, so we'll talk about that, and I think that's the only thing we are going to cover in today's lecture. So in between, I think this has been a one-way flow of information. Of course, at the end of the every session, we have had very interesting question answer session where we try to answer the queries from the participant. But today, because it's not that scientific a talk, it's more about good laboratory practice from an implementation point of view. I'll see if in between if I can ask you questions, and of course you cannot speak, but in a chat box you can try and answer. So let's see how does it go, and if it really works out, maybe in future class also I would like to maintain the same practice. So with that brief introduction of today's lecture, let me get started. So yesterday we have talked about drug development. Of course, from the discovery phase, in detail we have talked about how exactly you decide the target, how target is being identified, and after identification of the target, like an unmet medical need or a disease target, whatever you have identified, the next important step is you have to validate that target, and you have to validate using a of course an in vitro or in vivo assays, kind of a proof of studies, also proof of concept studies can be done to really validate your assay. And then we do hit identification through compound libraries. We run through a lot of softwares and look for those drug-like properties, and from a potency point of view, and from those hits we look for a lead. And then finally we have discussed in detail about how do we do lead optimization, what all studies are being done as a part of lead optimization. That's what we have discussed yesterday. So all this thing basically takes your molecule into preclinical development, and lead optimization majorly happens in preclinical stage. Okay, and once your molecule clears the preclinical testing, then only the potential molecule which are highly potent, which are safe, at least in non-clinical species, will proceed to clinical phase, clinical trials as phase 1, 2, and 3. So in preclinical testing, we have talked about animal handling. Again, in this particular picture, you can see a person is not wearing the gloves, and I would always recommend that please wear the protective wears. Okay, so apart from apron, you should be wearing gloves as well. So basically, preclinical, be it an efficacy study or a pharmacokinetic study or a toxicology study, you have to do the testing in animals. As long as it is a in vivo testing, you can do all this thing in an in vitro phase also, but there you don't use a live animal. Instead, we use either cell-based model or a non-cell based, maybe enzymatic model, or other models can be used in vitro setting. So what we discussed yesterday was an overview of drug development, and we discussed that before even we file an IND, what are the IND enabling studies, right? So what is the data package of IND looks like? So when you go to a regulatory agency to submit your data, what all data needs to be included in that package? That is what we have covered yesterday. So essentially, all the animal model studies for efficacy, the assay development, again either a cell-based or a non-cell-based assay for potency determination of your drug candidate or a series of chemically diverse molecules, and a PK/PD study. All this thing happens within preclinical scenario, and once you have all this data, then we go for IND filing. And as you can see, the chemical synthesis, one of the important factor is how you develop your formulation, right? So formulation development, it goes on all the way from preclinical till phase 1, 2. Those efforts are continuously in play during drug discovery, or especially during drug development. Okay, when in phase 1, dose escalation and initial PK studies are done, while phase 2, 3 are going on, PK/PD studies will continue to happen. Proof of concept also is being done here. Again, since it is happening in the patient's population, you can very well see the efficacy of your drug. And when you do a large efficacy trials, basically in large population of patients, from 1,000 or 5,000 numbers or more than that, you try and understand the further authenticate the efficacy of your drug molecule. Okay, so once you have all this thing data available with you, you can apply for an NDA application, to actually allow regulatory agency to register your compound, your drug, and further launch it into the market. So non-clinical efficacy, whenever we say, so where are those pharmacological studies are useful? So yesterday we have discussed that that will really help you in assessing an appropriate schedule. You really want to give a daily dosing, a weekly, or how you want to plan your dosing intervals, right? Also, it justification for a drug combination and understanding the effect at a molecular target. So when you do an efficacy study, these are the objectives you try to achieve. Both efficacy, I again and again I have emphasized on this, and again I would say, see, whatever efficacy you get in vivo or in vitro in a non-clinical studies, okay, in animal studies, may not dependably predict clinical efficacy. And that's the very reason even today many of the drug candidates fail in clinical phase, in different phases of clinical trial, phase 1, 2 and 3. And one of the major reason of their failure, as we have discussed earlier, is lack of efficacy. So they have exhibited very good potency. They seem to be highly efficacious in animal species, but when you go to human, in clinical trial, same efficacy may not be reproduced, and that's why many drugs fail in clinical trials. And there are reasons, as we have discussed earlier, because of heterogeneity of the disease. The same disease can have different expression in animals versus in human. There is definitely interspecies difference in terms of ADME, because the enzyme expressions could be slightly different. There could be polymorphism. So whatever data we generate in an animal species may not accurately predict human data, right? And then there's a role of immune system. Those immune system are also slightly different, more well defined in human. So you may find some rejections in acceptance of the drug by the body in human as well. So these are the few reasons. But again, as we have discussed earlier, all this thing is there, that's a fact, but still you can try your level best and minimize this differences or variables between non-clinical and clinical species. How do you do that? Is once you establish an in vitro metabolism, say for example in vitro hepatocytes or microsomes, try and choose a species which is showing very similar metabolism profile of your drug candidate with human hepatocytes or a human metabolic human microsomes. So what happens is, at least the variability because of difference in metabolism or because of expression of an enzyme will be minimal. So you can definitely take, as we have discussed in one of the classes, that nowadays there are humanized mouse models are available, where we try to have the disease expression very closely resembling to what we see in human. So that way you can get your efficacy data also as close as possible what it is going to be in human, even in non-clinical species. So there are means and ways how you can really minimize this variability, but by far failure of drug in clinic is majorly because of two reasons. One is lack of efficacy. The same efficacy as you see in non-clinical species is not reproduced there, and second reason is because of toxicity. So please remember that. So preclinical studies, especially we talked about ADME. So what is the objective when you do in detail we have discussed yesterday. So the objective here is to study the effect of test material with respect to absorption, distribution, metabolism and excretion. What body does to the drug, that is what we are trying to understand. Okay, so duration definitely could be few hours to days based on how long this compound can stay in your body, right? Initial pilot study, when you do with a lesser number of animals with lesser time points, you can really plan a detailed PK study. So we should plan our time points for the PK study in animal species in such a way that you can get a accurate or more relevant AUCs, the exact exposure of the drug in the body, as well as half-life. In other words, you should have time points which can cover during which the drug concentration in the body should be minimal, right? So that should be your deciding factor on duration of the PK study. And animals required, as I already mentioned, typically rodents and non-rodents you can pick up. We also talked about safety pharmacology. Again, what is the purpose there? That is to investigate undesirable pharmacological effects, right, of the test material. And usually they are single dose. Again, can be done in 2 species. And the core battery, what we try to cover, cardiovascular, respiratory, and CNS side effects. So that is about safety pharmacology, what we have talked. Then yesterday we also talked about toxicology study, right? So here, this is the summary, like when you dose an animal either in a single dose toxicity study or a repeated dose toxicity, eventually you can observe the animal for its behavior, for the clinical signs, for their body weight, for their feed intake, for any other mortality or morbidity. But apart from that, at the end of the experiment, be it 14 day, 28 day, 90 day, or 6 months long toxicity study, you're going to have samples in form of, first, as blood samples. Maybe you can take it just before the sacrifice, and those blood samples you can actually look for quantification of the drug, that how much concentration of the drug is there after the duration of toxicity study. Number one, you can look for some specific biomarkers from the same samples. You can also collect, of course it is part of the study, most of the toxicity study ends with collection of organs and tissues. So close to around 40 organs, like when you do a typical toxicity study and you collect the organs, I can tell you that except for the viscera, pretty much everything else is collected. 40 organs means almost everything from the body is being collected, including your eyeballs, to all the major organs, to specific glands, everything is collected. And you can really keep it in 10% formalin, and then finally you can do what do you say, pathology on that. So you can prepare the first, you can have a gross examination, look for any pathological lesions you can see, and then finally after the study is over, you can start preparing the histopathology slides of each of those tissues. So you can compare control versus the dose groups, low, mid, high group, or the recovery groups, and draw some conclusion out of it. At the same time, the blood samples which you collect, you can do hematology, biochemistry, clinical biochemistry can be studied in that. Look for enzymes, look for biomarkers from that. Drug I already mentioned, from the blood you can separate the plasma or serum. Again, from the tissues also, you can do a tissue distribution kind of thing. I mean, not exactly distribution, but the amount of drug available in different tissues at the end of the toxicity study, that would be drug analysis. This is typically how when a LCMSMS graph looks like when you do a quantification using LCMSMS. And some of the samples can be then stored for further analysis. So you see, like pretty much everything is used up from this animal. The only purpose of this is the design of the toxicology study is such that you understand even a minute untoward effect of a drug. Okay, so if at all there is any adverse effect, then we would like to capture that from this toxicology studies. Which are those? Either acute, subacute, chronic, subchronic or chronic toxicity studies. So that is how toxicology study design happens, and the endpoints are there. So this entire thing is a huge database actually, right? So that's why documentation is very important. And again, as it says, endpoints and timings are agent specific. It's very much true, as I said, based on the disease target which you select, which you're working on, based on the drug of our interest, the drug candidate which is under evaluation, you can really decide the endpoints. You can really vary those endpoints. Of course, there are guidelines, OECD guidelines for each of the toxicity studies, but still you can always have an additional time points or additional endpoints added to that study if at all you feel that is going to fetch you some more highly relevant information about toxic effects, potential toxic effect of your drug. Okay, so that's how toxicology study is being designed. So About the timing of the studies, like we have talked of different studies, I just thought let me put into perspective the timing and how which particular phase of the clinical study is supported with this. And there were a few questions about pediatric, so I have tried to include that information also within this table. That what all data is really essential to conduct a pediatric trial. So safety pharmacology generally 1 to 3 weeks depending on the pharmacokinetic data, and typically it has to be done before phase 1. It is a part of your IND documentation. Toxicokinetic, as I said, it's a typical pharmacokinetic study which you dose a drug to the animal either by oral or IV or any other route, intended route, typically which should be same as the route in your target species, right? And then you collect blood at different time points, we separate serum or plasma out of it, and then we quantify the samples for drug concentration. So when you do the same studies at toxicology doses, so along with the toxicity study, you maintain a separate satellite group where at same dose groups like low, mid, high, you dose them and then collect plasma samples at different time point. That study is called toxicokinetics, right? So typically up to 14 days toxicokinetic studies, right? Because it tries to mimic a toxicology study. And again, before phase 1, single dose toxicity study or dose escalation study, it can vary depending on what kind of effect you are seeing in terms of morbidity or mortality. The time duration can vary, and if there are any local tolerance studies which needs to be done. So all of this thing, the timing for this is before phase 1 clinical trial, and that data actually supports phase 1 and phase 2. If you talk of repeated dose toxicity study, which could be acute, subacute, chronic, or subchronic studies, as I already mentioned, it should equal or exceed the duration of phase 1 and 2 studies, right? So minimum 2 weeks is the time defined for this, and maximum is 12 months. So when you do a chronic study, it can be for 6 months to 1 year, and when you do an acute study, it can be minimum of 2 weeks, right? So generally 1 to 3 months for, if it is a biotech derived products, generally, of course, it depends on the product also, but generally it is 1 to 3 months long studies. Timing would be before phase 1 and before phase 3, right? And phase 1 to both, while it is going on, it really supports the this particular toxicology studies. And then genotox studies. So as you can see, before pediatric clinical trials, okay, for any pediatric clinical trial, before that you should have genotoxicity data in place. As I have already mentioned, that when you give the drug in pediatric neonates or young ones, newly born infants, it's very important that the safety profile has been completely thoroughly studied, and that data is very important. So genotoxicity is must, only then you can consider doing pediatric clinical trials, either phase 1 or 2, right? So reproductive toxicity studies, again, you can see complete all female reproductive toxicity and genotox studies before phase 1 and 2, correct? And pre and postnatal development studies before the marketing approval, because these are long-term studies, so it can be completed before the marketing approval. So, and all this thing should be actually completed before pediatric studies. So now you see pediatric studies are being given very much importance from a safety point of view, because if at all you have not studied the entire toxicology toxicological profile or potential of any adverse effects from your drug which may come, then that poses a big risk for pediatric clinical trials. So all this data typically has to be generated before pediatric clinical trials. Same way, carcinogenicity studies, again, before a pediatric clinical trial, this is very important. It really supports. If your compound passes carcinogenicity, genotoxicity, reproductive toxicity, then there are chances that you can really get permission to do pediatric trials as and when required. So that's about the different toxicology and toxicokinetic studies, when they are to be done, what is the probable duration, what is the timing, like before which clinical trial phase it should be completed, or along with which clinical phase it can be completed, and which clinical studies are supported. Now, that is what we have covered in this. Okay. So with that, I think we will start our today's topic, which is good laboratory practice, right? So let me just start with, so when we say good laboratory practice, if anybody would like to just type in the chat box, what do you understand by GLP? Okay, I mean anything, any words which comes to your mind about GLP. When you hear the word GLP, what comes in your mind first? If you can just type in chat box, we can take 5-10 seconds time, and then if you can respond, that would be great. Okay, I can see it says hygienic, it says avoid contamination. Good. Anything else? Accuracy. All right. Repeatability. Fine. Okay. Very good. So, see, I could see few responses, and then they're really highly relevant. Okay, so the moment you say GLP, I would say hygienic, avoid contamination. They are very specific to a particular study. But again, it's true. Somehow we can really correlate this to a good science, and yes, that is relevant. Accuracy of the data which you generate, it should be repeatable, absolutely relevant. And then I can see a complete definition. GLP is a quality system of management control for research laboratories and organizations to ensure uniformity, consistency, reliability, reproducibility, quality, and integrity of products in development for human or animal health. Okay, very nice. So this is typically the I can see a complete definition of GLP. So very good. So good to know that at least the term is known to all the participants, and most of the people are aware and they can correlate themselves to GLP. So very nice. So you see, GLP is nothing but it's a quality system. Correct. Let's take it as a quality system, and how we will cover it up. Okay. So whenever we hear GLP, the first thing which comes in mind is rules, safety, right? Standards, guidelines, qualification, quality. So these are the words, FDA. So these are the words which comes in our mind the moment any lab hears about GLP, right? So please remember, GLP is a FDA regulation, okay? And basically, how do you define GLP? It is a set of principles, okay, that provides a framework within which laboratory studies are planned, performed, monitored, reported, and archived. Right. So yesterday in detail we have seen that how well you can plan your study, how well you can actually execute the study, then how the quality check or quality assurance comes into the play. So how it is monitored. Finally, you have to really report the study, I mean the data, and then you have to archive. So as long as you have a framework, the quality system, as somebody has rightly mentioned, the quality system, as long as you have a quality system in place which can ensure that there is a framework within which a particular lab or a laboratory can really plan, perform, monitor, report, and archive the data or a study is called GLP. Okay, and sometimes people, whenever I have seen this thing many times when we visit colleges or labs, the moment you say GLP, people just show you that okay, we just wear all kind of protective wears like apron, face mask, gloves, goggles. So we really follow GLP. So please, friends, of course, it is one of the important aspect. Okay, safety, personal safety, it is very important, but only by wearing gloves, wearing protective wears doesn't really comply GLP. Okay, so that is of course important, but from a safety perspective. But GLP is not only restricted to your personal safety, correct? We will see GLP is basically it includes your existence, your systems of entire lab. Okay, so whatever system you have in place for operation of your lab comes within the purview of GLP. Okay, not only the entry exit procedure or not only wearing a protective gear wear. So that's a misconception about GLP. So please remember this. Okay, so OECD principles of good laboratory practices were revised in 1997. Okay, so what it says that principles of GLP should be applied to non-clinical safety testing of test items which are contained in a pharmaceutical product or in a pesticide product, cosmetic, veterinary drug, food additive, feed additives, or industrial chemicals, right? So whatever non-clinical safety testing is being done, their principles of GLP are directly applicable. Okay, so what is the purpose? The purpose is how this test item is like what is the purpose of testing? That this test item is to obtain data on their properties, on their safety with respect to human health and environment. So at the end of the day, whenever you say safety, so safety is in respect to the human health, because if human is the final target species, if it is animal, then it would be animal health and the environment. So we just want to ensure that this particular objective is met. So unless specifically exempted by national legislation, these principles of GLP apply to all the non-clinical health and environmental safety studies which are required by the regulation for the purpose of registering or licensing pharmaceuticals, pesticides, food, additive, cosmetic, veterinary drugs, similar products. So very important. You see, unless by a national regulatory agency, unless by a specific, if any country has that exemption, uh, otherwise, I mean, unless that exemption is there, principles of GLP are applied for registration purpose of almost everything from pharma, pesticides, food, feed, cosmetics, veterinary drugs, similar products, everything. So whenever you go for registration of this compounds or this particular products, your data on safety should have been generated in a GLP environment or as per the principles of GLP. So let's start from the history. Okay, when actually GLP came into play. So in 1976, I don't want to go into the detail, but just a few dates to remember that draft GLP came into play in 1976. Okay, in 78, GLP for non-clinical studies came into the play, and then there were a lot of different versions which has come through. Okay, and then directives and then inspection guidelines and all those things came in India. Important for us, 2002 was the year when National GLP Compliance Monitoring Authority came into India. Okay, came into play in India. And very importantly, until 2011, India was not a part of OECD countries. So India joins OECD agreement on mutual acceptance of chemical safety data. What does it mean? Whatever data is generated, suppose India being a member country, if the data is generated in India as per the GLP principles in a GLP certified lab, the safety data, it will be accepted by other OECD countries. Vice versa also is true. If it is generated in other countries, will also be accepted in India as long as safety is concerned. So these two dates you should remember. In India, this came into play in 2002, and in 2011, India became one of the OECD country member. So GLP is a formal regulation that was created long back, as I said, by FDA. Okay, although it is originated in US, but it has an impact in entire world. Okay, non-US companies that wanted to do business in United States, or register their pharmacies in United States, they have to comply with United States GLP regulations. Okay, so finally what happened, or non-US companies, finally they started implementing GLP in their own countries also. And in 1981, the organization OECD, what I call it as Organization for Economic Cooperation and Development, produced GLP principles, and that formed international standards. Okay, so OECD came into play in 1981. GLP was introduced by FDA in 1978. In India, it came in 2002, and in 2011, India became a OECD country member. So why actually GLP was created? Okay, the question is why it is required. Why we should really have a quality system in place? Can't we do better without those quality systems in place? So again, let me stop here and ask: what do you think? Why GLP is required? Or just answer me yes or no. Do you really think without GLP we can do good science? Or if you feel GLP
Excuse
is
me, sir,
required,
your
then
PPT
why
is not
GLP is
visible.
required?
Excuse me, sir, your PPT is not
Hello.
visible.
Yes, yes. So actually, I have asked a question here, so that's why I just come out of it. Yes.
Yeah, okay,
So
okay, okay.
can you just type in how many of you feel that it is required? Say yes or say no. GLP is required if you feel. You can type yes. If you feel it is no, you can write no. Okay. So I hear few yes here already. Okay. So see again. See, it is a personal perspective initially to start with. Now it is a regulation, so it is a must. Okay, there is no option. But still, if at all there is a doubt in your mind that we can live without GLP, I'm sure with my subsequent slide you will be convinced and you will agree that GLP is absolutely required. Okay, so why this GLP was created by FDA in early 70s in 76? Because they came to know through their investigation that poor laboratory practices were prevalent across United States. Okay, we always complain our country. We say that in India things are not done in a standardized manner, but it's not true. I mean, we also follow a much organized way of doing good science in India, and a lot of labs are GLP certified here. But there was a time, even in United States, in 70s, many labs were doing poor laboratory practices, and they actually carried out the investigation in 40 labs. And you know what they realized after investigating 40 different labs within USA, they discovered that a lot of fraudulent activities were going on. Just few examples: equipment not being calibrated to standard form, therefore giving wrong measurement. So very important. You see, they were using instruments, say for example spectrophotometer, say for example HPLCs or a mass spec, and those are not calibrated. So now once an instrument is not calibrated, means it is not going to give you an accurate reproducible data. It can be any instrument. So now if you use that particular instrument for any quantification or any analysis as a part of your study, safety study, or any other study, whatever data you get will not be reliable, right? Say for example, you go to a pathology lab, you give your blood sample, and they have got automated instruments with them. If those instruments are not calibrated, the data which you get from that, you can never believe that data, right? Simplest example, in your own college, in your own lab, you may be having a weighing balance, as simple as that. Or if you're a chemist, you like to weigh the compound. Imagine the weighing balance is not calibrated. So what happens? Weighing balance is supposed to tell you, of course, depending on what kind of a weighing balance you have, it can tell you how much mg accuracy of the weighing, right? Up to 2-digit decimal or 1 decimal, it can accurately weigh the compound or weigh whatever items you put on that. Suppose instead of you weigh 2 mg and your balance is not calibrated, and instead of 2 it shows you 1.5, whereas the weight is 2, but since it is not calibrated, since it is not maintained properly, it has been used continuously without calibration, now it is showing you less weight. For 2 mg it is showing you 1.5 mg. Imagine the implication. You will consider you have taken 1.5 mg, whereas You have actually weighed 2 mg. Now you prepare your stock solution. Now, whatever experiment you will do, you will consider your concentration as 1.5 mg per whatever stock solution you prepared, say per ml. But in reality, it was 2 mg. So basically, you will be off in all your experiments, right? So that's why it's just an example of a weighing balance, but it holds true for almost every instrument which we use. So what they found in these 40 labs was that equipments were not at all calibrated, okay? Inaccurate or incorrect accounts of actual lab study. They were doing lab studies, but they were not documented properly, right? So they do, like in your lab, if you do an experiment and you don't even record how much mgs you have weighed, what reaction you have done, what is the incubation period, and after 5 days you completely forget, and then you have to note it down in your rough notebook somewhere. So you see there is no traceability, and you may forget. Instead of 10 minutes of incubation, you may record 15 minutes incubation, right? Because you don't recollect. So incorrect or inaccurate accounts. And finally, inadequate test systems means the improper training of the people, study plan was not well designed, problems with recording the data, no standardized procedures. Like standardized procedures means if you say in your protocol, in your SOP that I'm going to centrifuge my blood sample for 3000 RPM for 15 minutes, means you have to follow that. You just can't do 3000 for 5 minutes just because you are getting late. You want to complete your study faster. So now you cannot just spin it for 10 minutes, whereas in your protocol it says 15 minutes. It's just an example. So like that, you have to stick very strictly to your study plan. Poor care of animals. What did we study when we talked about animals in last couple of classes? That animal is your test system, right? So it's very important that you maintain a controlled environment for your animal studies, right? They should be at ease. We have talked about enrichment. Why? Because we want them to be very comfortable. So whatever data we get is reliable. So if poor care of animal is there, they are not well fed, they are not given proper water or supply, they are not properly sterile animals to be used. I mean to say the hygienic conditions are not good, they are suffering from some infestation or infection, then their one or the other organ system is affected, and then the drug compound which you test on them, the data which you get will again not be reliable. So you see, these were all the prevalent practice in 40 labs which were investigated by FDA back in 70s. And the most famous example, I want all of you to really understand this, that one of the lab that went under such investigation out of those 40 labs, which came in a very big headline also, that lab was Industrial Biotest. Okay, the name of the lab was Industrial Biotest. It was a very big lab, okay, and it was an outsourcing lab. It was kind of a contract lab. So many big companies, just an example, Procter and Gamble, all of you have heard about. They used to outsource their studies, animal studies, to this lab called as Industrial Biotest. And what they realized in their investigation, that it was discovered that mice which they used to test cosmetics, as you know, for cosmetics one of the important testing is animal testing, you have to do. So for lotion and deodorants, you have to you have to do the irritation test also, right? So they checked in mice and they realized that the lotion and the deodorants which they apply were causing cancer, and the mice used to die, as you can see in the picture. It's a dead mice, right? So ideally, under GLP, as I said, documentation is very important, reporting is very important. Lab should have reported the same thing, but since no such practices were followed, what did they do? So Industrial Biotest lab, they threw all the dead mice, okay, and they covered the result, deeming the products good for human consumption. You see, such a big crime. So your mice have died just because you don't have a proper recording procedures in place. They simply threw those dead mice, replaced them with a healthy mice, and completed their study. And the outcome of the study was that the products are pretty safe for human consumption. So you see, such a big crime. So this was discovered during their FDA investigation, and then they actually what they did, they really investigated thoroughly all those who were involved in production, like manufacturing, distribution, and sales of for the lab. They eventually were sentenced for a jail term. So you see, this was the punishment they give. But the point I'm trying to make is, if you don't have proper GLP principles in place, it's not that every time you will be committing a crime of projecting or passing a compound or a drug which is failing, but even otherwise, when you try to do authentic experiments also, but if your documentation, if your lab procedures, if your training of people, if your documentation of chemicals, reagents used in the lab, all those things are not in place, if those procedures are not really custom, I mean to say not customized, they are not defined, if you don't have a defined quality assurance or quality check in place, then there is every chance that you may not be able to do good science, and thereby you may fail. And these are the implications of that. So you see, this compound, whatever lotion or deodorant, it would be detrimental to our health. So this actually led FDA to come up with GLP back in 1976. Okay, so what is the objective of GLP? I hope now all of you would have understood why GLP is important, right? So quality checks are important everywhere. Correct. See, we all study like 50 people. Let me take example of this class. Okay, we are really doing this training. Okay, one way is I can blindly believe that everyone, each one of you are really learning everything correctly, and you have learned everything. But that could be an assumption. There should be some quality check in place. So at the end of this, we are going to do an assessment, right? As Maheshwari has already discussed earlier, by the end of this training, we'll do an assessment. It is not to really put pressure on you, but just it's a quality check. It is just we want to assess, and then we want to see whether you can clear that assessment or not. If you clear that assessment, only then you qualify to get a certificate of the training. So in other words, I'm just trying to tell you that everything has to be well defined within the procedure. Okay, as long as every single procedures also in your lab are well defined, GLP is little tough. Okay, so the objective of GLP is GLP makes sure that data submitted are true reflection of the results that are obtained during the study. So we want to avoid any manipulation of the data. As you can see here, it was a Procter and Gamble example they have taken. But say example COVID-19 vaccine. So vaccine, you see now those vaccines, me or you or everyone will be taking those vaccines. So we want that data generated should really reflect the results of the study, right? When they've done the clinical trial, I hope, and I'm sure it is as per GCP, good clinical practices. The safety data is earlier in clinical, non-clinical should be as per GLP, and only then the data which they have obtained is reliable. So now, if that vaccine is taken by all of us, we will be safe, right? Because all that data would have been already generated. So that is the importance of GLP. That is the objective. Another important objective is traceability. Whether data is traceable or not, that is what GLP also ensures. And it promotes international acceptance of the test. As I already mentioned, it is not possible every time to keep on testing, doing same experiments in different countries. If there are international standards well defined, then what happens is, as I mentioned, whatever data generated here should be acceptable in other OECD member countries also, right? And it should be audited. As I said, a quality assurance monitoring is most important thing. So now you see, objectives are very well defined for GLP. So within the principles of GLP, what all things are really within the purview of that? So first and foremost important thing, what we'll cover all of that: test facility, organization, and personal, right? The institute, the lab where you want to do the test is test facility management. All the scientists, people working will qualify as personal. The test systems, right? Like an animal facility, so that becomes a test system. Like an analytical lab, that become a test system. The facility, what all facilities you have, apparatus, material, reagents, right? Quality assurance programs, performance of the study, reporting of the study results, standard operating procedures, SOPs, personal and test facility organization, already we have covered, and finally archiving of records and material. So in nutshell, principles of GLP are applicable in all these aspects. So pretty much it covers everything about an organization. Yeah. So what do you call a good science? Okay. So what is a good science? Basically, we say good science means we do a good experimental design, right? With proper parameter with defining proper parameters of our experiment, of our acceptance criterias, endpoints. So if you define an experiment, you would like to have endpoints, parameters well defined, right? It should be purely based on scientific procedures, correct? If you want to call your experiment as a scientific, we would definitely want it to be based on a scientific procedures. Of course, we want to control the variables, correct? So say for example a clinical trial, say for COVID-19 vaccine trial, so we will try to cover the variables. So how do you do that? You try to have as many variables included within the scope of your study, right? When you do a clinical trial, you try to cover larger population, so you're trying to cover as much variables as possible by increasing the size, right, of your clinical trial. And different, say we include different age groups, we include different area specific people, we include different disease conditions, say for COVID, different symptoms exhibited by different stages of patients diagnosed with COVID. So based on that, you can really include as many variables as possible, right? So control of variables, most important. You want a logical, scientific interpretation of the results, correct? And results, they should be accepted by everyone scientifically. So this is what we say good science. So as long as your particular experiment or your project aim or goal revolves around this, we can always say it's a good science. And GLP, forget about everything. GLP just helps you in doing good science. So if you want to really do all this thing, you want to achieve all this, GLP really helps you in doing that. Okay. So it starts with good organization. Okay. So what is a good organization? We have talked about good science. What is a good organization? Means you should have a facilities, required facility in place. You want to have qualified personals in place, right? You should have proper resources, planning in place for the studies, definitions of responsibilities and trainings of personnel, control of records, and verification process implemented, correct? So within GLP, to be a good organization, you want to have all this thing. And we will see how do you really consider a qualified personnel. Means doesn't mean that with the best resume in place a person becomes a qualified person. A qualified person by education, by training, and by his experience. So this all three, and by training, so all this thing has to be documented. We will see how that happens. So that really makes a person qualified. It's not just verbal qualification, it is a documented proof which really proves that a person is qualified. Same way, planning of resources and studies. So all that plans, the study plan, the annual maintenance plan, annual calibration plan for your instruments, all that thing should be in place, which will define the planning of resources, right? definition of responsibility. So each and every person will have a definite role to play in a GLP environment. Okay. So it really makes sure many a times we work with less manpower. We try to do multitasking. Sometimes it is good, but when you in a GLP environment, we try and avoid to do that, right? Multitasking is good, but we should never really compromise the quality of the work to be done. So that way, the GLP organogram is really defined. We'll talk about it later. There should be a complete record control over the records. It's not that whatever data has been generated, the report has been made, anybody and everyone can access that. So there should be a procedure in place for keeping records of the studies. Okay, and there should be procedure to gain access to that. And then verification process. So quality assurance should be there. So quality assurance, again, we'll see, will be at different places. The quality assurance can check your protocols, can check your SOPs, can check your documented procedures. There could be in-process audit where they can see how a particular process is being carried out. Say you're doing a study. If you're following a protocol, then that process can be audited as per the protocol you had planned, right? So somebody can just check the protocol and see if you are doing your experiment as a scientist, are you abiding by that protocol which you have prepared and approved? So that can be an in-process audit, correct? So that can be done. So that is a verification process implemented, correct? So personal, so as I said, in terms of good organization, these are the things covered. Let's talk about personal. So I already mentioned this. So for individual person, hold on a second. Yes, organogram. So one of the most important thing in GLP is every organization. You can have it in your lab, also in your college, university. Please have a well-defined organogram in place. Who is the head of the institute? Under him, which all departments are there, under that which all study persons are there. Of course, for a GLP organogram it is different. We will see in a subsequent slide. The head of the organization is called as test facility management. Under him there will be the study director, the person who is responsible for executing the study, and that study director will have different support from different departments like an analytical scientist or a veterinary scientist or a pharmacology scientist. All those people, or a pathologist, all of them would be a support system for a study director. Then there will be a separate archivist whose only job is to archive whatever data, whatever test items comes to him. Then there is a quality assurance. So this test facility management should have a separate reporting, direct reporting of archivists, study directors. You can have one or more study directors, as many as you want to have, and the quality assurance unit, they should be directly reporting to test facility management. So that entire organogram should be in place. And also please remember, GLP gives very much importance to individual quality control as well. So what happens is, suppose if somebody is on leave, okay, suppose archivist is on leave, what you do? Suppose you need to archive the documents. In his absence, you can't do it, right? So GLP always recommends that you should have one backup person identified for each job role. Say for example, test facility management, then you should have a deputy TFM, right? So in absence of TFM, deputy TFM can execute the work. Archivist, there should be a backup for him who can act as an archivist in his absence. You have a quality assurance, a backup for that, a study director. So like that, say for TICO, like a test item control officer, so a deputy TICO who will really receive the drug, who will dispatch the drug to the concerned department. Say a toxicology study you want to do, you need 100 mg of the drug. So you cannot just go and take it on your own. You have to put a request, a written request to TICO, that is test item control officer, and he will really approve that, okay, you can take this drug, and he will issue the drug to you. He will take your signature on that. So there should be a deputy TICO. So you see, it is very well defined role for every individual in an organization if your lab wants to qualify as a GLP lab. So you should have an organogram. Each individual, his job description should be there in place. Now you see how important it is. If job role is not defined, many a times people are exploited in universities, in academia, or in a private industry. So each one should have documented, well defined job description. Okay, what he or she is supposed to do. Their curriculum vitae should be attached, which is a standard format. Not that I prepare 6 page of resume, my colleague will prepare 15 page of resume. There should be a well-defined format of a curriculum vitae approved by quality assurance unit QA. Okay, and in that format each individual in the lab should have their curriculum vitae defined. So you see, trying to bring about uniformity and training records. Suppose see, many times it happens, why this certificate is required for all of you after this training. Why that certificate which will be issued to you by Andhra Pradesh State Skill Development Corporation is important, because that certificate authenticates that you have undergone this training. Correct. Without that, verbally you can say anything. You can say you have done 1 month, or you can say 10 months of training, right? You can say 8 hours a day I was doing training, but what is the proof of that? So that training record is just an example. Like that, any training you undergo, you should have that records, and that record should be documented in a file. Why I'm saying is, when any auditor comes to audit your facility for a GLP thing, this is what they look for. When they ask for a personal file, they would expect an organogram in that. They would expect every individual's job description in that. They want each one of curriculum vitae in a uniform format, and they want their training records. So a quality assurance person has undergone any training, virtual, in person, or any on the job training, or a webinar or a seminar, that particular certificate would be in that file. So when an auditor looks at that, that really proves that these people have well defined job description, they have a required experience, qualification, and they have required training records in place, right? So this is the records you need to keep from personal point of view. In terms of responsibilities, these are the few important aspects of any GLP facility. As I mentioned, the head of the institute of a lab would be called as test facility management. So he is the final authority. So you may call him anything, CEO, MD, or anything, but from a GLP perspective, the person should be test facility management. And also you should have a deputy TFM who can work, who can sign the documents in absence of TFM. To execute any study, okay, if you want to do an acute study, subacute, subchronic, chronic toxicity studies, one study director should be there. Okay, so of course a study director can manage more studies. That is doable based on how much of a space you have, how much of a time engagement a particular study director have. But you should have a well-defined studies allocated to different study directors. Okay, so for one particular study, this gentleman or this lady would be responsible, and he will be called as study director. So you see, so well-defined responsibilities are there in GLP. Then there'll be one principal investigator. Okay, so study director can actually appoint a principal investigator for his study, and there'll be other support study personnel who could be scientists or colleagues supporting him. They'll qualify as study personals. So all those things should be defined in a study plan. So you see how well organized it is. When you define your study plan, you should have all these people mentioned in the study plan. So related to that particular study, this would be the people responsible, and responsibility of each of these people would be well defined in that. So anything related to that study director would be responsible. The final report would be signed by study director and countersigned by TFM after quality assurance certificate issuing. So then that report becomes a final report. So you see, so well defined plans are there in place for GLP, for operation of any lab. For a quality assurance program, I just mentioned that you should have well defined quality assurance program in your lab. Okay, so there should be a QAU unit itself and with direct reporting to TFM. They should have entire plan in place that we need to do this many audits, we need to do this many inspections. So that is really required for a quality assurance unit program. Okay, so it's not that they can just walk into any study and start doing audit. That entire plan should be in place, that okay, these are the studies we are going to audit, these are the inspections we have planned, and TFM should really give them permission to do so. Okay, and they should listen, they should review, and they should report regarding the fact. So this is about when they are doing an audit. This is your internal quality assurance program. Okay, so don't confuse it with an outside auditor. When an outside auditor for a GLP study, when he comes from an FDA, that is from a regulatory perspective. This is your internal quality assurance system. So if you have very strong internal quality assurance unit in place, then rest assured you can clear any audits from any regulatory agency. So that all depends on your internal quality assurance programs. Okay, so when internal auditors come for any audit, they should just listen, they should review how you're doing the procedures. Are you complying with the protocol or the SOP of that particular activity? And they have to report the fact. Okay, so they can do a study audit, as I'm mentioning, while dosing or blood collection. Any of this study, in study audit they can do, any of the in process audit they can do, and they can simply come and do a facility audit. What is facility audit? They may come and look at the entry exit procedures of your lab. Okay, do you have that SOP in place? If you say that a person comes in a lab, first he has to put his biometric finger, I mean do a biometric attendance, and then he enters in. If that is your SOP, they just come and check that. If anyone, if they find coming directly inside without doing biometric, means it's a finding by your quality assurance unit, right? If you say that in your protocol entry exit protocol you have defined that they have to come and they have to sign a document for their attendance, then they need to do that. If they don't do, means it's a finding, correct? So all the things, they come to your lab and they would look at an instrument, they look for a calibration record. If calibration record dates are correct, it's fine. If calibration is not done, then it's a finding, correct? So it really ensures that the entire quality system is in reality implemented in the lab. So that is the role. So quality assurance unit should be a separate unit with direct reporting to TFM, and they should be doing in study, in process, and facility inspections. Okay. When they come for study audit, what they look for? They look for a study plan. They look for your procedures. They look for the final report. In a final report, is the final report covering all the aspects which you have mentioned in the study plan? While study plan, they would like to see, is it, if you have mentioned as per any guideline, did you follow that guideline, right? Is it really approved by quality assurance unit? So those are the things which they look for. When they come for in-study audit, they look for procedures. As I already given an example of centrifugation. If you are defined certain time, say let's take another example. We will say incubation. So if you have mentioned an incubation time of 30 minutes for a procedure, then that's what they look for. If you do it for 20 minutes, that's a deviation from your study plan. So that is the audit your internal quality assurance team will do. For facilities, as I already mentioned, all the equipments, informatic systems, training, documentation, all these aspects comes under facilities. So let's move on. We talked about good organization, facilities. So size, construction, location of your facility, right? Minimize alterations. Do you have different activities being done in the same facility, but at least physically separating each, right? You cannot have a quality assurance unit, HR, your scientists, everybody within the same room, correct? It can be a biased thing. So say for example, you do an animal experimentation. So animal facility should be separate, correct? Your HR HR is okay, fine, based on the space availability, but an archivist. So you should have a area where archiving really happens, correct? Where you can control the archiving procedure. So that should be segregated. You should have a separate place for quality assurance because they will be reviewing the SOPs, protocols, documents. So you should have a separate space for them. You should have a separate office space for your people, right? Not everybody can sit in your lab. So these are all the facilities points which you should keep in mind while you really design your facilities. Okay. Now let's consider what are the points to consider. So experimental point of view, test systems, right? Study types, numbers. So you should have that entire list in place that, okay, in 1 month, we are planning to do 10 studies. Each study will have a specific study title. Each study will have a specific study number and well-defined rooms, if you have, where the studies will be carried out. So it avoids any confusion, any what do you call any mishandling or confusion in terms of mixing of different animals from different studies. So all those confusions it tries to avoid, right? So you should have a well-defined plan in place and an entire list with title of the study and the study number. So when auditors, internal or outside auditors come, they can really trace back a particular study by its study number, and that study number would be used as a reference everywhere. Suppose you go and use a weighing balance, so there will be a weighing balance logbook, so you want to make an entry into that. So please give a reference of a study number that I weighed this compound this much mg and for this study number. So now it is recorded. You go for animal issuing. Say you want to do an acute toxicity study, so you want to issue male female animals, you're given a requisition. So in that also you have to put study title and study number. So now you see at the end of the experiment all these documents will come in same file. So traceability will be from study number. So from the time your drug candidate was issued by a test item control officer to you, animal issuance form, weighing of the drug, everywhere there will be that study number. So that study can be traced completely after months of completion of the study if you have that system in place. You got the point? In terms of personal security and welfare, so do you have all the protective wears in place? You're handling animals without gloves, without apron, without face mask, not acceptable. That welfare, as I mentioned, did you really vaccinate your people who are handling the animals for tetanus, for hepatitis? That's a good welfare practice, right? So all those things should be kept in mind while you think of your facility. Operation. Now access, cleaning, storage, maintenance, waste disposal, there should be procedure for each of this activity, right? Sometimes you may not want access of everyone to a particular area, so that should be access control. It should be well defined, and in that area except for the designated people, nobody else should have an access. For cleaning, it should be well defined. It should be, the frequency should be recorded. If you say as per CPCC guideline we want to do disinfection of our system at least twice a day, then it has to be documented somewhere that twice a day it is being done. Then is it really recorded? So that's how the individual activities, the operational activities has to be well defined. So there will be practically no activity happening in your facility which is not documented. Even, as per the GLP, generally for cleaning, like say for example, cleaning is done by people with lower qualifications, so they may not understand English sometimes. So it is said that you should have an SOP defining their activity, what they're doing in local language. Say for Andhra, it will be in Telugu, so that people who are doing the cleaning in your animal facility, okay, or who are doing washing of your cages or racks, who are not highly qualified, but still they should be trained to do their job. And for their easy access, the SOP of cleaning or washing, you should have written in English as well as in local language. So at least they can read Telugu and they can follow the instructions. So it is that well organized facility expectation in terms of GLP. And as you may realize, if you follow all the steps which we have discussed so far, in a way you're making your lab, you're making your institute very organized. Even outside of GLP, if you follow all this thing, I can guarantee that you would definitely be able to do good science. Okay. So in facilities, okay, it is again right. So test systems. So test systems includes well-defined procedures for everything. Within the test system comes biological, which could be your animals, the test, the drug, cosmetics, agrochemical, veterinary drug, which you are testing, that chemical, physical, or combination of those, all those becomes test systems. So you should have a very well-defined test systems, right? As I mentioned, animals, bacteria, cellular, subcellular organs, all this thing. Maintenance, as long as instruments are there, their maintenance should be well defined. So it's just that you should have some document or some procedure in place that we are doing an annual maintenance of the instrument. This is the contract we have signed with the agency, and once in a year they come and do the maintenance, and after they do the maintenance they actually put a label that maintenance was done on this date, or a calibration was done on this date. That's it. This is all they ask for. So now you see, if I have to go and visit a lab, and if I see an instrument, and if I see a maintenance label on that, calibration label on that with correct date and frequency mentioned, I'm fine that okay, this is. And if I ask for a file of last maintenance record, there should be a document which states that the maintenance was done in this way and this was the acceptance criteria. So I know that this instrument is perfectly in a working conditions. You should definitely follow national standards. Like for animals, you should simply follow CPCSA guidelines. That's all. There is no separate guidelines for GLP for that. As long as auditor come and see if you are complying with the national standards, that's all is required. That is qualifying for GLP. Okay, all the routine procedures, cleaning, water, diet, environmental parameters. I said temperature, like plus or minus 23 degrees Celsius. But fine, you should have a form out there. You should have a thermohygrometer which will test the temperature. You should have recording being done. So somebody who should be measuring the temperature, either it is automated or manually, somebody should note that temperature. So the guy who is noting, he could be biased. So some superior should sign that document on a daily basis. So once that daily basis pages are piled up, you can file it for a month record of your temperature, humidity. So you see, everything is so well defined. So an auditor comes and he looks at that file. He can look for 10 days prior, morning 10 o'clock, this was the temperature, evening 10 o'clock, this was the temperature. And why I'm saying morning, evening? Because you will have an SOP in place for monitoring or recording of temperature in a animal facility already in place, where you have defined that temperature monitoring will be done in at 10 o'clock and evening 5 o'clock. So you have, and as per the format attached, that's it. So as long as you have it in place, auditors are so happy that you have a protocol in place which says how to record the temperature or relative humidity. You have a form where the data entry goes, and you have a file where it is all maintained on a regular basis with signatures of your supervisor or the quality assurance people. So you clear that audit, right? So all this documentation is what GLP insists for. And I'm sure with our discussion, you all will agree that all this documentation in a way is really helping you in maintaining your particular lab in a better way, right? Irrespective, forget about GLP, even otherwise you follow these principles, I'm sure our instruments are well calibrated, they are well maintained, our people are well trained, all the records are being maintained, the facility is as such well maintained, all the job descriptions are really defined. In your organization will really do good, right? So even otherwise you're going to do well if you follow principles of GLP. Okay, so housing, again, assignment to treatment groups. This is in particular to the animal facility. So assignment to treatment groups, where are you going to house them, right? Where is the cage location? You should label them. Where you discard your animals, that area should be well defined. How do you housing, like a reception where the animals are going to be received? You should have a quarantine room. Interestingly, earlier many people were not knowing the meaning of quarantine. Thanks to COVID-19, I think majority of the people worldwide know what is the meaning of quarantine. So you should have a quarantine room in your animal facility where you keep. Suppose you are not doing in-house breeding, you bring animals from outside. You can keep in this room so that they get acclimatized to this new environmental condition, and then they can be shifted in with other animals in different rooms. So you should have that in place. The raw data, regular checking of the raw data, all that thing should be in place. Let's talk about SOP. One of the important aspects of GLP is SOP. What is SOP? Standard operating procedures, right? So if there is a very simple definition of an SOP. Write what you do what you write. That's it. It is so very simple. And that makes up majority of your GLP thing. Write what you do and do what is written. Means every single procedure in your lab, from your entry exit procedure to the operation of an instrument to handling of any material to receiving the test items to archiving procedures for reporting, every procedure should be well defined. There should be a document which explains, why it is important, because this makes the system self-driven. Many a times in our college, in our university, in our non-GLP labs, the systems are not self-driven. They are driven by individual person. In other words, if I'm doing an experiment, I'll have all the documentation or everything in my mind. And suppose if I don't come tomorrow, then experiment will not happen. Why? Because I've not documented, and that particular activity is completely dependent on me, and which is very detrimental for any organization or any lab. So the idea here is, if you have this SOP procedures in place, if you try to document every single activity as an operational menu for your instruments, for your activities, for your procedures, what happens is, now it is a system driven. Tomorrow if I'm absent, somebody else can come, read the document, read the SOP and do the same procedure, or they can operate the same instrument the way it has to be operated, and still they'll be able to get the results, still they'll be able to get the reproducible results data, and that is the purpose of SOP. Okay, so SOP defines how to carry out protocol specific activities. They are most often written in a chronological listing of action steps. So basically, I always say that in when I was studying in 12th, we usually have that flowchart preparation, right? And the most basic example was that how do you prepare tea? Can you explain it in a flowchart manner? So we used to say that the first put on your gas, light up the gas, put a container on that, pour water into it, allow it to boil, or pour tea bags or tea into it, allow it to boil. So it was all flowchart, step by procedure which we used to draw. That's exactly what you have to do in SOP, right? In a chronological listing, just mention any activity. That switch on the mains, switch on the individual units of an HPLC, the chronological order you have to mention. Check for the mobile phase bottles, fill the mobile phase bottles, you choose the column, open the column compartment, fit the column, open your what do you call auto sampler unit. So like that you can define every activity. So you, as minutely you define, that easy it is for anybody else to reproduce that activity, right? So they are written to explain how the procedures are supposed to work. So routine inspection, cleaning, maintenance, testing, calibration, every activity should have an SOP. Also, an SOP should capture one of the important things. Suppose if it is related to an equipment, what action to be taken if it fails. So if it is defined there, that's it. It closes the loop. Means you are able to operate an instrument if anything fails, any routine thing. Of course, if an engineer intervention is needed, you can't do anything about it. But a routine troubleshoot, you can always make it a part of your SOP. Even analytical methods, raw data, keeping a record, reporting, storage, mixing, retrieval of data, all the activities of your lab can be converted into a standard operating procedure. Finally, statistical procedures for data evaluation. So for a good science, we always say that data which we generate out of animal studies or a human clinical trial should be statistically significant, right? So statistical procedures are not simply chosen from a textbook. They are biostatisticians, right? They adopt certain standards which are deemed acceptable within that particular field. Okay, it's not that every time we take an ANOVA table and try and do a statistical significance. There should be a justification, and it's not that after looking at the data, after looking at the results, you decide which statistical tool to use. That's wrong. That is what is not acceptable. You should define the statistical procedure to be used in your study plan. Means I'm doing this study, I'm having this many groups of animal, I'm having this many groups of human volunteers, this many arms of the study, number of human or number of animals, and this is the test. I'm going to just an example like a Z test or a T test or whatever test, DUNET test, which particular statistical test I will be doing. So you have to even define your statistical procedure in your study plan. You cannot really decide the statistical tool after looking at the data. Otherwise, it is definitely biased. So that is what regulatory agency look for, whether there is a statistical acceptance statistical procedures in place or not. As I mentioned, for instruments, each instrument should have validation record, right? So the process necessary for any analytical laboratory. One thing I talked is calibration, another is validation. So data produced by faulty instruments may give appearance of a valid data. So you have to define frequency of calibration, that we'll be doing calibration once a month or once in 3 months, validation once a year, whatever you have to well define, because it varies from instrument to instrument. So for each instrument, you have to well define that. Okay, whenever an instrument performance is outside the control limit, you should report, and it must be discontinued. It's not that you should throw away the instrument, but keep a label that out of calibration or out of validation. So somebody who is using that instrument, if he sees that label, he may not use that instrument. So you can avoid getting any faulty data. So in equipment record, so there should be a complete file for your instrument, where it should start with an index page where each instrument should be enlisted there. So what all should it include? Name of equipment, the manufacturer, the model type for identification, serial number of the instrument, date the equipment was received in your lab, and copy of any manufacturer's operating instructions. Okay, and then in any case, next to the instrument you should have the SOP, that is how to really operate that instrument. Okay, for reagents and materials, the policy is to ensure that reagents used are specified in the standard operating procedure. Okay, this is very important. I'll spend few seconds on this because this is very common, and I have seen it most of the time in especially in colleges. I see those reagent bottles. I understand you're talking about many students are coming, the student flow is there, many reagents, chemicals are coming and going. But even then, we should have a policy in place where each of this reagent or chemical bottle should have a label. Okay, and the label should have the date. It should have the name of the chemical or a reagent. It should have, suppose if it's a bottle, it should have a date when it was opened, and there should be an expiry date, right? This is very important, because without that, there's no way somebody can really make out. Because over a period of time, say in chemistry lab, I see that because these chemicals are corrosive and all, many a times even the label, the original bottle labels of reagents and materials are chemicals are really not even visible. So nobody knows when this was bought, what is the expiry. So at least whatever chemical reagent bottles you have in your lab have this bare minimal label in place, which defines the name of the reagent, date when it was opened and the expiry. Or if it is a bottle which you have prepared with combination of some reagents yourself, then it should have the composition of that, and you should put the expiry. So that is must in your lab. So when your internal quality assurance people come and audit, they will look for that, and when an outside agency comes to visit your lab, they can always see those labels. Okay. Purchasing and testing should be handled by a quality assurance program. Perfect. So you should have purchasing and testing records also, right? You can, if you want to use it for a longer period, you should prove that it is still usable. It should be labeled. Deteriorated, outdated reagent solution shall not be used. Include the date open. And the storage condition is also very important. I forgot to mention, apart from date of open expiration, you should have the storage condition. Sometimes they are to be maintained in fridge conditions, 4 to 8 degrees, and they are kept in room temperature. How does anybody even know that the storage conditions are different? So if storage conditions are defined, not you but somebody else who can notice that can put it at the right place. So these are the procedures which really makes your operations within your lab more organized, more optimal, conducive conditions in your lab to carry out any scientific experiment. Okay, analyst. So some acceptable proof of satisfactory training. This is a very specific example I'll take of a analytical chemist or a bioanalytical scientist. So there should be some procedure to really authenticate that a person is able to do a particular task. Say for example, there is a procedure defined that this is how he has to prepare the calibration. So you have to make him do that, report that data. There should be some historical data against which it can be compared, and that particular thing can be documented. That yes, although his experience is in microbiology, but he is a trained, he's trained for bioanalysis, and this was the experiment done, and this is his training record. So that's all. Because not every time you can have a specific qualification person doing a specific job. Sometimes we can't afford to have so many people. So we do multitasking. So all you need to do is you create those records, and which proves that the person is trained to do the job he is doing outside of his qualification. Right? If you have sufficient people, as I said, as soon as possible. Laboratory normally done by an external agency. Of course, we do like ISO. Those are all what do you call OSHAs for safety and all. Laboratory certifications. Few labs you will suppose you go for an NABL. You would like to have an NABL accreditation. So all this thing can be done. Okay, but in nutshell, you should make sure that you got adequate space, your good ventilation, a storage space, and overall hygiene conditions you are maintaining in a GLP lab, non-clinical lab. So species sample, you can say it can vary among the laboratories, but it should contain, there should be a connection between a set of analytical data and the specimen from which they are what obtained. Yes. So when you archive the samples, when you archive the raw data, try and see the correlation. So when you run as I mentioned, just an example that you mentioned study number everywhere. So when you have analytical data coming out of it, even if you have mentioned a study number, that study number can be traced back to the actual sample it belongs to. Or you can have a specific sample name which can be mentioned on the analytical data, each and every of your graph, which is also mentioned on the sample, which is archived. So there is a good tracking system in place. Okay. Documentation, maintenance of all records provide documentation which may be required in event of legal challenges. That's the reason many a times patent litigations are going on. So maintaining all these records anyway helps you in that. There should be general guidelines followed in regulatory laboratory to maintain record for at least 5 years. So from a GLP perspective, it says that you should be able to maintain the records minimum for 5 years after completion of the and now there are separate archive facilities also available, because not every lab can have so much of an archive space where they can store data up to 5 years, right? So length of the time over which laboratory record should be maintained can also vary with the situation. So it's between you and the sponsor, you can decide. Coming to properties of an archive. Now, finally, the last step is you want to archive the data, raw data, study data, final report, study plan, study deviations, amendments, everything you want to archive. So your archive should be, you should have protection against robbery, right? Against fire, you should have fire extinguishers in place, avoid water pipes so that there is no leakage and it can lead to further messy situation in your archive. Pest control barriers so that they cannot eat up your records if they're kept in open in archive. Air conditioning systems to maintain environmental conditions, and adjacent room for query the documents. Means if you don't have a sitting space out there, at least an adjacent room where somebody can really take those documents and then review that and then keep it back. Of course, there should be a procedure to issue the document and then put it back. So you should have an ideal archive, should have all these properties in place. So these are the questions asked by an analytical by any auditor for an analytical instrument. Very basic questions. What is this equipment used for? Is the instrument within the specification? And is the documentation to prove this available? If it is a calibration, is the label over there? Okay, within that specification range. If the instrument is not within the specifications, what action has been taken to overcome the defect? Can the standards used to test and calibrate the standards be traced back to national standards? Very important, because many times we do the calibrations ourselves, right? To a weighing balance, say a weighing balance. So there are standard weighing balance weights available, weights. So you can use that. So now those standard weights, are they as per national standards? Were they really as NABL certified weights? Only then we can use internally to do the our weighing balance calibration on a daily basis. So these are the things asked from a perspective of an instrument. A typical study plan, if I talk about, we already covered that everything should be covered in a study plan. This is a typical study plan in a GLP environment. First and foremost, it should be approved by study director. You should have a reference of the guideline which you are following. You should have description of the most relevant process of the study. You should have detailed procedure defined in that. There should be a schedule. If it is going to happen in a phased manner or one after the other activity is going to happen, you have to put a schedule of that. There should be an identification. Each study should have a specific study number. Identify the data for study, test, reference purpose, persons involved. There should be an identification. You should definitely have a title and the objective. Okay, a description of the test and the reference compound, information of the test facility and the sponsor, who are the study director and other responsible person, specific dates of doing the experiment, detailed test system, experimental design, and any amendments if involved. So you see, all this thing should be included in a study plan. So you can imagine, if you prepare such a detailed study plan, chances of expecting any untowards results is bare minimal. Even if you get anything, the data would be really authentic. Let's not talk about good or bad data. Whatever data you get out of such a detailed study, it would be easily traceable, it would be authentic, it can be monitored, and it can be reported. Okay, so we have talked about study plan. Let's talk about what a final report should include. Again, the study identification of the study, basically the study title and the study number, information regarding the sponsor of the test facility, actual dates. This is very important. A statement from quality assurance. A quality assurance should put a statement that this is as per the principles of GLP or not, right? Description, results, and then the archiving procedure. That where it will be archived. So these are the content of a final report. So let's understand. Now we have understood entire principles of good laboratory practice, from a facility, test system, test items, personal, study personals, instrumentation, facilities, everything, how a study plan looks like, how a study report looks like, what all things should be there within the lab. Let's see what happens if a workplace does not comply with FDA's good laboratory practice standards. If an auditor comes and if it doesn't qualify, then generally it can be a disqualification of a facility. So before a workplace can experience the consequence of non-compliance, First of all, they ask for straight away. They cannot just come and disqualify your facility. They'll ask for an explanation for this disqualification, why it has happened. So states the purpose of disqualification as the exclusion of a test facility from completing laboratory studies or starting any new studies. Okay, so you have to give justification. If that justification is satisfactory, you have still chance that they may not disqualify. But if they are not really convinced, then they will disqualify, and they will say that this particular test facility should be excluded from completing the ongoing studies or starting any new studies uh, because they are disqualified in our audit. So what are the possible violations labs do? And this is a very common thing. Many labs have been penalized by FDA. Falsifying the information for permit registration, any required records, falsifying the information relating to the testing protocols, ingredient, observations, failure to prepare, retain or submit written records required by the law. So these are all the violations which have been reported from time to time again for the regulatory agency. So what are the grounds when actually your facility can be disqualified? So a testing facility which fails to comply with one or more regulation implemented as per the GLP, you can be disqualified. Your lab can be disqualified. Failure to comply led if failure to comply led to the adverse outcome of the data, right? If it affects the validity of the study itself, then take it for writing, your lab would be disqualified. Or warnings or rejections of previous studies have not been adequate to improve the facility's compliance. Suppose previously some warnings or rejections for your particular studies have been issued, and those are very serious, then also it's a ground for them to disqualify your facility. So let's understand what happens if your facility is disqualified. What are the consequences? So the FDA states that the commissioner will send a written proposal of disqualification will be sent to your facility. Okay, a regulatory hearing on disqualification will be scheduled. If commissioner finds after the hearing the facility has complied, then a written statement with an explanation of the termination of disqualification can be sent to the facility. And if disqualification did not affect the integrity and outcome of the study or did not occur at all, then the same study may be reinstated. So there are chances, you still have a chance. As I said, you have to give a detailed explanation, and through that, if you can prove that the integrity and the outcome of the study has not been impacted, then also they can give you a permission to continue with your studies by the commissioner. And of course, it is not a permanent disqualification. A commissioner can definitely inspect the facility again and determine if they need to reinstate the facility, and they can remove the disqualification. Okay. So at the end of it, I think we are almost on time. I would say that in your free time, please go through all these guidelines like OECD, www.oecd.org, and you will find all the guidelines of OECD. You should also look for Indian, because we operate in India, so National GLP Compliance Monitoring Authority guideline, website www.indiaglp.gov.in. For ICH guidelines, please follow www.ich.org. For European Regulatory Agency EMA, it is www.ema.euro, and for USA FDA, you have to visit www.fda.gov. So with these sites, you will see hundreds of documents are there, and where all your lab, your kind of work for your college or university falls in place, you should look for those guidelines, right? But these are most popular guidelines for anyone to learn, especially pharmacy students who are doing the quality assurance. You should really, or pharmacovigilance and all, please review this guidelines. Like ICH are most widely accepted guidelines, OECD are most widely accepted guidelines, and either your product can be registered in USA, then it's FDA, or in Europe, then it is EMEA. Same way for Japan and for Brazil. Different countries have different regulatory agencies, but these are most popular one. That's why I mentioned here. And for Indian GLP, go for www.indiaglp.com. So with that, we come to the end of our today's session on GLP, and I will be more than happy to answer any questions if you may have, or else we will conclude here and then we will again meet up tomorrow at around 11 o'clock. Thank you. Maheshwari, if there are no questions, then there is only one question which was remaining for me, which I wanted to discuss. But maybe in tomorrow's class, we will discuss about the impact of kidney failure, chronic kidney failure, on ADMA properties. Okay, especially on the clearance. We will discuss in tomorrow's class when we take up a new topic. But if any other we can really, is it possible for you to show the live SOP in experimental labs on animals? Okay, so there's a question. It says, if is it possible for you to show the live SOP in experimental labs on animals? Oh yes, of course. I can show. Maybe okay. So I don't know what you understand by live SOP, but I can surely share an SOP on an experimental animal. We have entire preclinical research lab where the training and the actual experimentation on animal happens. So we have SOPs for each of those processes. So what I can do is tomorrow when we start our class, what I'll do is I will display the SOP. Okay, or let me see if there's any soft copy of the SOPs. I can actually display that and show you how an SOP looks like. Yeah, that's a good question. I can definitely do that. So tomorrow I can show you example of a few SOPs for usual procedures in animal facility.
Thank you, sir. Students, anyone having any queries, please put in your chat box. Please respond.
All right. So, if no questions, then we would conclude the class and we would reconvene again tomorrow at 11 o'clock and again try to cover what all topics are still remaining from drug discovery and development. But I hope and wish that we should be able to finish within this week, and then maybe we'll do some revision or recap next week, and then finally we can have an assessment on the last class. So that would be the plan. So Maheshwari, just one question for you. Within this week, how many more classes we have? Okay, your line is
Three
muted.
classes is there, sir.
How many days?
Three, sir.
Three more classes.
Yeah.
So we have tomorrow, day after, up to Friday. Oh no, up to Saturday you have classes?
No, 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 sir. Up to 12th we have section.
No, no, that is fine. In this week, how many
2 days.
classes we have?
2 days.
2 more days?
2 more days. Thursday,
Okay,
Friday. And again
fantastic.
Monday we will have.
Sure, sure, definitely. So,
And
I'll try to, yeah, I'll try to complete within one class if possible, and then remaining two class we can do a revision so that we can do assessment in the last class.
yeah, on 12th we will have assessment.
Sure.
Yeah.
All right. So, okay, before I conclude, just one question has popped up here in the chat box. What is the diet of food for experimental animals in labs? Okay, so there are vendors who actually, uh, in every state, the feed is available. But basically, for experimental animals, we give pelleted feed. Okay, so they are in form of pellets, and there is a fixed percentage of protein, carbohydrates. So they are well-defined rodent feed. They're called as rodent pellets. Like our feed we have different components as per our requirement in terms of protein, carbohydrate, fat. Same way, their feed are also manufactured accordingly, and pelleted feed is what is preferred by laboratory animals, and it is available. So there is a rat feed, rodent pelleted feed, a rabbit feed is available. So different for hamster, the feed is available. So for different animals, those feeds are available, and it can be procured. But please remember, all this feed and all, they are generally, they should be sterile, okay? Because we don't want to just feed them anything which is non-sterile, because your facility is sterile. So anything which we use, we tend to, even the water which we use should be sterile, so as to avoid any infections within the colony of the animals, experimental animals you're having. So many colleges I see, they themselves make their pallets. You yourself can make those pallets, as long as you maintain, depending on, for growth, you should have more of protein percentage. Suppose it's a diabetic model, you would like to have more of a fat, carbohydrate into your diet. So like that, you can define your feed. But pelleted feed is what is accepted by experimental animals. We have another question: how we know the duration of dosing on animals in preclinical trials? Yes, you don't know, definitely. So you need to do a pilot study, right? You would have done an in vitro experimental study, in vitro metabolism study. So we'll have some idea about the half life, correct? That from metabolism perspective, what is a half life? And you will be doing one pilot PK study, and based on that data, you can really define the duration of the dosing. I mean, duration of the dosing once you do your pilot PK study, correct?
Yeah, thank you, sir. Students, anyone having any more questions, please respond. Otherwise, we'll close the section. Please put in your thought box.
All right, so no more questions. Then let me thank you, all of you, for being there with me for entire 2 hours, and looking forward to connect with all of you tomorrow. And in next class or so, we'll complete the entire course. And in last 1 or 2 classes, we'll do a quick recap of entire discussion so far, what we've done in close to around 12 days. And the last class, we'll have the assessment. So thank you all of you.
Thank you, sir. Thank you so much. Students, I have given the feedback form in YouTube, Facebook, and Airmeet also. Please submit. Based on that only, we'll provide the certificates. And at the end of the day, we'll have a assessment for you. Based on that, we'll provide certificates by grade-wise certificates to all of you. Please check it. Okay. Don't forget to give the feedback form. Okay. Thank you. Thank you, everyone. The